Let's take a look at the results pages for this lab. So we're starting here with the experiment A, control test tubes. And I put in here what should be filled out on this worksheet right here. So on the control experiment, the test tube number 1 was supposed to have starch solution as a substrate. Indicator was iodine, and that's the familiar color change to dark blue, and that is a positive control. The second test tube was supposed to test if iodine reacts also with glucose, and so the glucose is the substrate, iodine is the indicator, and there should be no color change. So the iodine, of course, has this amber kind of golden color to it, and since there's no change, this is a negative control. Water and iodine, let's see if that gives any reaction. That is, of course, also a negative control. So water and iodine is supposed to be no change, no color reaction, so negative control. Number 4 is testing glucose and its reaction with Benedict's reagent. If you do boil it up, then the sky blue color of the Benedict's will turn, in the presence of glucose, will turn to orange. And if it's a lot of glucose, then it's going to turn into this rusty red kind of precipitation. And that is a positive control. The reaction of water with Benedict's, there is no reaction. That is should give you no color change even if you're boiling it up, and that's a negative control. There were 2 more control test tubes, and that was to test the effect of the enzyme amylase. So you are adding starch and amylase into test tube number 7, and starch, no amylase, just water instead, and to test tube number 6. And then, of course, in the beginning of the experiment, you have starch present, of course, so you get a dark blue color. I'm sorry, here in the beginning of the experiment, both test tubes should be dark blue. Then, of course, the test tube that has the enzyme in it, the enzyme will digest the starch away, and so after incubation, the test tube should have no more blue color. It should be this amber color from the iodine only, because there's no more starch present, versus the test tube that didn't have any enzyme. Test tube number 6 is still dark blue because the starch is still there. So that means test tube number 6, starch is still there. Test tube number 7, the starch was digested away with the amylase. And the Benedict's reaction should be negative for test tube number 6 because there should be no sugar should have been produced. There's no enzyme, so you get no product. And test tube number 7 is a positive control. That one, the Benedict's reaction should be positive. So that means the amylase digested the starch, and now you have a lot of sugar in your test tube, and so the Benedict's reaction is strongly positive. I indicated this here with 4+. Here is what this should have looked like if you would have done this in the lab yourself. You can see here 3 test tubes on this first panel right here. You can see the 3 test tubes here. Just have Benedict's reagent inside of it. Before boiling, you can see the sky blue color here in all 3 test tubes, showing the Benedict's reagent. After boiling, you can see in the first test tube it's turning red, and that means there was a lot of sugar in this test tube, and the Benedict's reagent reacted with it. Test tube number 2, the one in the middle right here, you can see there was no sugar in the test tube, and so after boiling, the blue color from the Benedict's reagent is retained. There was no color change, so that would be a negative control right here. So there was no sugar in the test tube. And this test tube number here, the last one, the 3, is showing a strong positive reaction. And so here is sort of the color scheme that sort of you're supposed to indicate. If the test tube turns red, it's like a 4+, and orange is like 3+, and it's sort of the relative amount of sugar that was in the test tube giving you a positive reaction. If the test tube turns kind of greenish, there was just a little bit of sugar in there. If it turns yellow, then it's a little bit more, and if it's orange, like this one right here, it was quite a bit of sugar in the test tube, and red means that it was the most, but these are just relative amounts. On our control reactions, this would be like test tube number 6, and this would be test tube number 7. So going back to the handout. So here, test tube number 6 was a negative control. That was just showing that without the presence of amylase, we really don't get any sugar production. and the Benedict's reaction would be negative. And on test tube number 7, if you have 3 milliliters of amylase and you provide good conditions, then you get a lot of sugar production, and that's here these 4+. Moving on to experiment B, there we're testing first the effect of varying concentrations of amylase on the amylase activity. So the hypothesis should be that if you have more enzyme, then you're going to end up with more product. And so you're testing in 4 test tubes. You're testing 0, 1 milliliter, 2 milliliter, and 3 milliliters of the amylase solution, that contains the enzyme. And we have the same amount of starch, 5 milliliters in all 4 test tubes. And then we're going to let this sit for an hour at 37 degrees Celsius and watch what happens. So, of course, if you have no enzyme, then you get no product. So that means the iodine result should be strongly positive because the starch is still there. The Benedict's result should be negative because there should be no sugar production. If you don't have enzyme, you don't get any product. And so test tube number 2 with 1 milliliter of amylase should give you a little bit less of an iodine reaction, because the 1 milliliters of amylase, they will digest some of the sugar, some of the starch away, and you will get some sugar. So you get a positive Benedict's result, maybe in brackets, or maybe 1+. And the iodine reaction should be still positive though, because there should be still some starch left. In test tube number 3, we have 2 milliliters of amylase, and that will give you a good amount of sugar. So you should have about a 2+ on the Benedict's result. And the iodine result, depends on how hot it is in the lab, but sometimes we have no more starch present, so then the iodine result is negative. Sometimes you have a little bit, depends again on the temperature in the lab a little bit. And the amount of amylase here, 3 milliliters in test tube number 4, that's full strength, and that gives you full amount of sugar production. So 4+ on the Benedict's result. All the starch should be digested away to sugar, and so that's what you see the 4+ here for. And the iodine result should be negative. There should be no sugar left. So the interpretation of the result means 3 milliliters of amylase gives you the best results, and in general, more enzyme means more product. So next we're testing the effect of temperature on amylase activity. Hypothesis is that, well, since amylase is an enzyme that works in the human body, then body temperature should be ideal. So 37 degrees Celsius, that's a good hypothesis, should be the best temperature. So we have 5 test tubes. Test tube number 1 is our negative control, as we had before. Test tube number 4, same as in the previous experiment, but it's ordered different here because of the temperature. So we're testing 3 milliliters of amylase on ice incubation. Then we have 3 milliliters of amylase at room temperature at about 20 degrees Celsius. Test tube number 4 was at 37 degrees Celsius. That is at the incubator. And test tube number 7 was boiled. The amylase was boiled. Now, amylase is an enzyme. It's a protein. And when you boil it, it denatures. It doesn't work at all anymore. So expected results. Of course, if you have no enzyme, you get no sugar production here in this case, and that means the iodine result should be positive and the Benedict's result should be negative. When you're incubating the amylase on ice, you get very, very little activity. It's not 0, but it's very little. So the iodine result should be positive, and the Benedict's result slightly, maybe a little bit positive. Okay, that's this one right here. Test tube number 6. Room temperature works better than on ice for sure. So you might still have a little bit of blue in your test tube, so it might be slightly positive, the iodine result, but you should definitely get a strong, good sugar reaction with Benedict's, and that's the 2+ right here. Test tube number 4, we already saw that. You get a negative iodine reaction, no more starch present, but you get a very strong sugar reaction. It means all the starch was digested to sugar, and now we have a 4+ Benedict's reaction. And when you're boiling the enzyme again at 100 degrees Celsius, this enzyme will denature, and it should give you no reaction anymore. So that means the iodine result should be positive, and the Benedict's result should be negative. There should be no sugar production because this enzyme is dead, literally. It's denatured. So the interpretation of results is that amylase works best at 37 degrees Celsius. The last part of this experiment tests the effect of varying pH on amylase activity. So we have 3 test tubes in this part. Test tube number 1, as before, no amylase, 5 mLs of starch at a pH of about 7. Test tube number 4, as before, 3 mLs of amylase and 5 milliliters of starch, pH about 7. And test tube number 8 has 3 mLs of amylase, 5 mLs of starch, but we're dropping the pH down to at least about 5 by adding some 2% hydrochloric acid. That's this stuff right here, 2% hydrochloric acid. And the iodine result after incubation should be positive for test tube number 1 and no sugar production because no enzyme gives you no results. So that's as before. Test tube number 4 gives you optimal results. We have 4+ on the Benedict's result. So massive sugar production, but no more starch left, so negative iodine color. And the test tube number 8 with the wrong pH. This is clearly the wrong pH for this enzyme. So the same 3 milliliters only give you moderate sugar production, maybe even just 1+. And the iodine result is still kind of positive because you still have some starch left. So the wrong pH definitely slowed down this enzyme. So the conclusion is that amylase works best at around pH 7, right around neutrality, which is what we should expect because that's what you would have in the small intestine where amylase works. Also, we have a salivary amylase that works in your mouth, but the one and that one also likes more of a neutral pH. So were our hypotheses correct? Yeah, I guess we made some good hypotheses. So they've all panned out to be true. What conclusions can you make? What's the optimal concentration, temperature, and pH? So the conclusion is that the best concentration is the 3 milliliters per 5 milliliters of starch solution in our test tube. So that works best. The best temperature was the 37 degrees Celsius in the incubator. That's the body temperature, and the best pH for amylase. Whether that is the salivary amylase or the pancreatic amylase, either way it likes a more neutral pH, and if you make it too acidic then amylase doesn't work that well. So best pH is right around pH 7. And what evidence would we use to make these conclusions? We used the starch test, so the presence of starch still, and the development of sugar. And so let me switch over here. Your row of test tubes may have looked something like this. So up here, in the first row, so up here, you can see the iodine reaction. So that's this row right here. You can see in test tube number 1 a very strong iodine reaction. So there was starch in this test tube clearly, and it's all blue right here. The iodine reaction is positive. So here in test tube number 2, you can see some turbidity. Maybe there was no iodine added just yet. It does look like as if there's starch in there, but hard to tell. So it's definitely, if it looks like this, then there was no iodine or there's no starch in the test tube. And here 4 is definitely positive on the starch test with iodine, and here clearly in the last test tube, remember here number 8, clearly a very strong iodine reaction with starch. So that's what it looks like when starch and iodine combine. They form this very dark blue color complex. The lower row right here, this row right here, that will be Benedict's test. Benedict's has a sky blue color right here. And in test tube number 1, you can tell this is a completely negative sugar test. So the Benedict solution did not change color, and it looks just as before. And then here in test tubes 2, 3, 4, 5 through 8, you have various amounts of sugar production. You can see in test tube number 4 clearly there's a lot of sugar production, and so very strongly positive Benedict's reaction. And so in the other test tubes you have various amounts of simple sugars present. Now let's take a look at experiment C. The emulsification of fat. What type of mixture is formed with the salad oil, water, and the bile salts? So that's an emulsion. And if you would have done this experiment, you can see under the microscope, you can see that when you are mixing, even without emulsifier, if you're mixing the oil and the water by shaking the test tube vigorously, you'll see tiny little droplets. So that doesn't change. But then without the emulsifier, the fat droplets combine and they get larger and larger. And we're going to take a look at that and continue with this in the next recording.